If you are someone struggling to find a matching organ donor for you or for someone you love, well, we have good news. The healthcare sector is about to change forever with engineered organs being on high demand. The concept of organ transplantation is not a new one because it has been around for centuries, but successful transplantations only became a reality by the 20th century, with the majority of recipients living for more than 5 years. But still, there is one big problem. The demand for the organs usually exceeds the supply, because of which the cost of the organs and organ transplants are extremely high. If you were to find a solution to this problem right after your basic economics class, you would think of manufacturing more products to meet the demand. But we are talking about organs here, not something that can be manufactured in a factory. Or is it? Because it looks like scientists have cracked that code by developing engineered organs. So keep watch till the end to know more about how engineered organs will change the medical industry forever. Most people who went through something like an organ transplant live a pretty normal life, but that does not mean that organ transplants cannot lead to any other medical problems. Some patients in the long term deal with problems like diabetes, high cholesterol, high blood pressure, gastrointestinal problems, anxiety, and depression. Some might also develop sexual problems such as a decreased sex drive or loss of function, but that problem only comes after the entire procedure. What raises more eyebrows has to do with one of the biggest challenges when it comes to organ transplantation, which is finding the right donor. When a person receives an organ from a donor, the body's immune system will start doing its work and will recognize the organ as a foreign body. Your body's immune system is designed in such a way that it protects you from substances that may be harmful, like germs, poisons, or even cancerous cells. These harmful substances have proteins known as antigens on their surface. As soon as these antigens enter your body, the immune system recognizes that they are not from the person's body, will consider them as foreign objects, and will attack them because they are different from what is produced in the recipient's body. Mismatched organs can trigger a blood transfusion reaction or transplant rejection. So to prevent this from happening, doctors match both the organ donor and the recipient by choosing someone with similar antigens to reduce the likelihood of the organs getting rejected. But the truth is, the match is not usually perfect, as no two people are similar except for identical twins. So doctors generally resort to using medicines to suppress the recipient's immune system, because the goal here is to prevent the immune system from attacking the newly transplanted organ when the organ is not very closely matched. If medicines are not used, the body will immediately launch an immune response and, like we mentioned earlier, will destroy the foreign tissue. But again, there are certain exceptions, like in the case with cornea transplants, because the cornea does not have any blood supply. As a result, transplants from one identical to another are almost never rejected. But not everybody is lucky to have an identical twin, because of which scientists need to look out for alternatives, and that's how engineered organs come into the picture. Before we talk about engineered organs, we need to talk about tissue engineering. This is a field that evolved from the field of biomaterials development, and it refers to the practice of combining scaffolds, cells, and biologically active molecules into functional tissues. The goal of tissue engineering is to assemble functional constructs to restore, maintain, or improve damaged tissues, or even some cases the whole. Tissue engineering emerged as a promising approach with two major goals. The first one is to develop tissue and organ substitutes for clinical transplantation to replace damaged regions and restore organ function. The second one is to build human tissue chips and replace animal models for drug screening and disease modeling. Up until now, multiple levels of complexity have been achieved with existing technologies to precisely position cells at scales from the single cell to a whole tissue level architecture. Each organ varies in its unique structural components like cell types, matrix, and architecture, along with the biophysical environment, which includes pressure and flow, and biochemical stimuli, which includes oxygen tension, cytokines, and growth factors to support these specific organ functions. The concept behind tissue engineering is not rocket science. The patient's stem cells will be grown in the laboratory, then these cells will be added to a scaffold material, and finally you have a laboratory-grown organ. But it's not that simple, and the technology is still at its infancy. Scientific studies related to healthcare are mostly about bringing some novel breakthrough treatments to patients. But things are always not that easy, because there are several experiments that need to be repeated multiple times to turn something that was cooked up in the laboratory into a viable clinical option for patients that suffer from several medical conditions that require an organ transplantation. Next, you need to look at regenerative medicine, which is a broad field that includes tissue engineering but also incorporates research on self-healing, where the body will be using its own methods to heal with the help of foreign biological material to recreate cells and rebuild tissues and organs. Terms like regenerative medicine and tissue engineering have become largely interchangeable as the healthcare industry focuses on cure instead of treatment for complex and often chronic diseases. So how does tissue engineering and regenerative medicine work? Well, you might know by now that cells are known as the built-in blocks of tissues, and tissues are the basic unit of function in the body. Groups of cells make and secrete their own support structures called the extracellular matrix. This matrix, also known as scaffold, does more than just support the cells. It also acts as a relay station for several signaling molecules. As a result, the cells will receive messages from many sources that are available from the local environment. Each signal can start a chain of response that determines what happens to the cell. By understanding how individual cells respond to the signal and interact with the environment and organize into tissues and organisms, researchers have finally cracked the code to manipulate these processes to mend damaged tissues or even create new ones. This process often begins with building a scaffold from a wide set of possible sources, from proteins to plastics. Once the scaffolds are created, the cells, with or without a cocktail of growth factors, can now be introduced. When provided with the right environment, the tissue will develop. The scaffolds, the cells, and the growth factors are all mixed together, allowing the tissue to self-assemble. Then there's the other method that uses an existing scaffold. The cells of a donor organ are stripped, and the remaining collagen scaffold is used to grow new tissue. This process is used to bioengineer organs like heart, liver, lungs, and kidney tissue. This approach holds great value because of the fact that scaffolding from human tissue that is being discarded during surgery can be used by combining it with patients' own cells to make customized organs that would not be rejected by the immune system. So how does both regenerative medicine and tissue engineering fit into the current medical practices that we have today? Well, when you look at tissue engineering, it only plays a very small role in the patient's treatment. So far, supplemental bladders, skin grafts, small arteries, cartilages, and even a full-size trachea have been implanted in patients, but these procedures are not cost-effective by any means. It's a different story for more complex organs like organ tissues like lungs, heart, and liver tissue, even those they have been successfully recreated in the lab. Yet there's still a long way for it to be usable and ready to implant it into an actual patient's body. These tissues for now are quite useful in any research related to engineered organs and also for drug development, because by using functioning human tissue, researchers could speed up development process and would also be able to give us vital information for facilitating personalized medicine, along with saving a lot of money and reducing number of animals that are used for research. So what exactly does the future look like for engineered organs or tissues? Right now, the biggest barrier to making engineered organs a reality is to get all of the cell types working together in a coordinated fashion, followed by scaling the engineered tissue into a clinically useful dimension. Most of the areas of tissue engineering already suffer from the problem of scaling. It might be easier to conduct tests on the scale of small rodents, but when it comes to humans, the story is different, as it is a lot more challenging. The biggest hurdle is the funding for such projects, because there is a huge deficit in funding, which is the reason why we still are not able to test engineered organs on human beings. But one thing is clear: patients need the help of scientists who continue to search for novel treatments and to finally crush the demand and supply gap of organ donors and recipients, and also fix all the issues that plague organ transplantation as a whole. Let's hope that the adequate funding will be provided for research to turn these ideas into a reality.